Let's start with a quick statistics. Do you know how many people died of cancer in the last year? It's 8.8 million people. And it's 1 in 6 deaths due to cancer. Unfortunately, this is going to rise by about 70% over the next 2 decades. There are certain limitations of the major treatment options against cancer, like chemotherapy and radiotherapy, which affect not only the cancerous tissue, but also the normal tissue or body part of the cells. Surgery and other available medications are not successful enough to fight the poor prognosis of cancer. But scientists don't give up, and they always try to find some new hopes. And to understand that, we have to go a little bit deeper into the cell. For example, if we go inside the colon cancer cell here, we'll see a bunch of reactions are going on there. But importantly, some of these genes from the DNA code for mRNAs, and mRNAs code for proteins, and which is actually major players or functions in the cell. And it's always, this process is called the central dogma of life. And it's found that some of these proteins are overexpressed or produced only in cancer cells, but not in the normal sense. So, and if we can block these proteins, we can probably kill the cancer cells. But many of these proteins are also involved in the normal cellular functions, and blocking them is not always wise. So we have to keep search for new useful targets against cancer. And for that, interestingly, we found that there are several other type of RNA molecules found in the cell that cannot code proteins. They can be linear or circular, and this is called non-coding RNAs. And these non-coding RNAs actually found to be around 98% of the RNA population in the cell, and very little is known about their functions. And often they are called the dark matter of the genome. So in my project, I decided to target these circular non-coding RNAs to selectively kill cancer cells. And for that, I designed specific genomic tool, and I insert this genomic tool into a genetically engineered lentivirus, which transfers this tool into the cancer cells, and eventually degrades the circular RNAs. And we measure, after breakdown of 5,000 of the circular RNAs, which breakdown causes the cancer cell death. So for that, we found that, amazingly, some of these targets really kill cancer cells. For example, one of these actually killed 52% of the cancer cells in a colorectal cancer cell model. So from that, we expect this work will lead to the new era of cancer therapeutics, like precision medicine and personalized medicine, and will also help us understand the dark matter of the genome in the future. Thank you.